So Dr. Wolfgang Pernice is an associate research scientist at Columbia University's Department of Neurology. He is also living with CMT, as are several members of his family. His research combines pathobiology, molecular medicine, and machine learning to advance rare disease genomics. Today, we asked Wolfgang to moderate a panel as a patient scientist that will discuss the importance of genetic confirmation of your CMT type. We know it's an issue. We hear from people all the time. You know, many people have a reluctance to get tested. So Wolfgang is going to have a conversation about, you know, what are the processes? What are the implications? What should I think about as I decide whether to get genetically tested or not? So thank you, and thank you for being here.
Okay, thank you so much for the opportunity to moderate this amazing panel. I can even see the panelists. This is so unusual that the moderator isn't in the room. My apologies for it. Unfortunately, I caught a virus, so I can't be there in person, which I really regret. I know these events are amazing, but I am fortunate to be able to use technology to link in and to still run this panel. Hopefully everybody can hear me. Just for orientation, what we're going to do is I'm going to start this panel off with a few slides on CMT genetics, and then we're going to join into the panel and discuss some more pragmatic questions around how to get genetic testing and what it means for individuals to do so. So I'm going to try and share my screen. Let's see whether the technology works out here as well. Can anybody see? Maybe Nick? Good. I see a thumbs up. Perfect. That's super helpful. Okay, so the importance of genetic testing. Last year we had a fantastic panel that I was fortunate to co-moderate as well with some of the most leading and accomplished scientists in CMT biology and genetics. And we were really fortunate to be able to make, I believe everybody in the audience from that day will agree, a strong case for why genetic testing is important. But our perspective was really more from a research perspective. You know, why is genetic testing important from a research perspective? This time we want to focus more on the patient story. What does it mean for a patient to seek genetic testing? But before we get into this, I want to go through what CMT genetics really looks like for a little bit to orient everybody. My laboratory at Columbia focuses on genomic medicine, on CMT and mitochondrial diseases. And what we try to do there is to close something that I describe as the cycle of genomic medicine. You'll be familiar with the idea, or to no surprise, what we would like to do in the clinic ideally is to see a patient in the clinic, go through genetic diagnostics, move that into translational research, such that we might be able to study what the disease mechanisms are in the lab and use those insights to translate back into clinical trials, a therapeutic approach that will hopefully benefit the patients. Along this aspirational cycle of genomic medicine, of course, we face many roadblocks. And one of the first roadblocks is right here in genetic diagnostics. You may have heard already, and unfortunately this is still largely true, that we are only successful about 50% of the time when we try to find a genetic diagnosis for a CMT patient. And what this slide is really to bring across is that without finding a genetic diagnosis, we really can't get started as a research community and as a biomedical research community in making progress and understanding what the disease is about and move towards the development of treatments. So genetic diagnosis, genetic testing is really the precondition for any research to be done in this way. Now, what I wanted to focus on today is the importance of patients themselves in moving this circle forward. And I have some slides that hopefully will communicate this message. First, this slide is unfortunately always a little depressing. Still, I feel everybody probably knows this. Around 1 in 2,500 people in the US have CMT. We estimate around 130,000 or so patients should be in the US with CMT. We already know 100 different genes, maybe more. And still the success rate that I just mentioned of genetic sequencing and testing is around 50%. So how is that possible? we see that, it differs a little bit or quite substantially by the different various subtypes. So if you have CMT, if you have a demyelinating form of CMT, we're much more successful than if you have axonal types. But nevertheless, our success rates, especially for these non-demyelinating forms, are kind of terrible. And I want to tell you a little bit about what's really behind this. Why is it so difficult to find these diagnoses? And what I've drawn out here is really something very simple. Science in general is really mostly about comparing groups. If we want to find out what the genetic cause for a group of CMT is, then we assemble a group of healthy individuals and compare that to a group of people with that disease, in this case, maybe CMT. And we sequence them all. And if we compare individuals against each other, we'll see many differences in their DNA, many different mutations. But what we're looking for is the one common denominator between all the patients that unifies them and differentiates them genetically from the controls. And if we have large patient groups and large healthy individual groups, then that's not so difficult. We can find the common denominator because we have lots of people. But of course, CMT is a rare disease. And so our groups of people are not that large. And when the groups of people are not that large, there will be many more mutations that could potentially be the common denominator in these patients. And now we have to distinguish lots of different mutations and determine which ones of those are benign. So benign mutations are the kinds of variation, the kinds of genetic variation that determines, our hair color or how tall we are and so on. But what we're really looking for is, of course, the pathogenic mutation that causes our disease. And what happens in these scenarios in which we can't find a single one, but only have a couple of candidate mutations, is that we in science then have to work through prioritizing which of those variants might be the one that we should invest into following up. And then we can use various bioinformatic tools in order to try and understand which of these mutations is more likely to be the cause of CMT. Now in the age of AI, there are many more powerful tools than we had available only a few years ago. But still, most of the time, what happens in these scenarios is that we aren't able to prioritize between some of these mutations. And what happens then is that the process simply stops before we can move into experimental validation. And one of the reasons for this is that despite the fact that we have done a lot of sequencing in the Human Genome Project, most of our efforts into understanding human genetics really focus on only a small number of genes. You can see here, this dark gray line outlines the number of publications that are available for this TP53, not a CMT gene, just a gene that the research community has really studied a lot. And the point here is that our CMT genes are typically in this gray mess down here. They're not very well studied. And this is also true for many genes throughout the human genome that we might want to be interested in. And then if there's not a lot of information about those genes out there, we can't reason about whether that gene might be likely to cause CMT. And that means that in many of those scenarios, we're unable to properly prioritize between candidate mutations that we have identified in CMT sequencing of CMT patients. And we simply don't proceed into the expensive and time-consuming experimental validation stage. So what can we do about it? You won't be surprised that my answer to this is more patients. If we had more people getting genetically tested, then we have larger groups that we can compare. And again, the pattern recognition, finding the common denominator is easier, and we can move from the identification of one single mutation straight into the translational and mechanistic research. Patients participating in research is not only important for genetic testing, but for all of the stages of the genomic medicine cycle that I've outlined before. And that's why it's so great to have the CMTRF assemble you and many other patient organizations across the world, assemble many other patients to make CMT research and genetics really a community effort. So the message of this part of the panel is really patients and their families plus doctors together. We're strong and we can make strides. And now that I've hopefully made the point that genetic testing is important for us to gain better insights from the research perspective, I now want to start into the panel. And I want to introduce, let's see if I can see you guys. I want to introduce first Trisha Buell, who is a genetic counselor at MGH and works with CMT patients and helps them understand their genetic test results. Say hi. Perfect. And Nicole Faulkner, who is, I believe, the medical director at Invitae, one of the genetic testing companies that helps us, meaning CMT patients, also clinicians and researchers, understand CMT genetics a little better. So I'm going to stop sharing this for now, I think. There we go. And I'm going to start with the first question for the panelists. So both of you work in different capacities in CMT genetics. And I would like to have both of you maybe introduce how you work with CMT and tell us a little bit about your work. Maybe Trisha goes first.
So, hello, good afternoon, I think it is. So I am a genetic counselor and clinical research program manager. I have worked in the field for about 18 years now, and but in terms of neurology and genetic counseling, much more recently with CMT. And it has already kind of been mentioned. I love working with patients and related to why or why not an individual might be interested in seeking genetic testing. What are the potential benefits? What might be the potential drawbacks? And deciding really on an individual basis if that is right for them.
I'm Nicole Faulkner. Welcome to Boston. I'm so excited to see a lot of people here. I'm born and bred in Massachusetts, so super proud. And have met a lot of people from surprisingly far away yesterday and today. I did raise my hand yesterday when they said, are you a patient or are you a academic or are you industry? So I am industry. But I'm a bit of a horse of a different color. We heard a lot from people with therapeutics yesterday. So you have CMT, what is your hope? What is your future? What can we do for you? I am a service provider that works at the other end. I work with your clinician, your clinical team, genetic counselors, usually as you're on part of your diagnostic journey. And we heard yesterday that that can be kind of long and arduous. I do not have a child with CMT, but I do have a child with a chronic medical condition. So I can speak to being a parent going on a diagnostic odyssey, and it can be really difficult. So today I'm doing a non-branded talk. I do work for a company, but I counted up last night in between clinical rotations, companies that hired me and then companies that bought them and companies that bought them. I work for 10 different diagnostic companies. So I will try to give a broad perspective. And I do hope I know Wolfgang sent us questions, but I hope we'll get to a Q&A. If not, I'm here all day, you're here all day, and we're here for you. So happy to answer any questions.
Amazing. Thank you for those introductions. So I just tried to make the point that from a research perspective, it's important to do genetic testing. But as I said, for this panel, we want to dig into a little bit more of the pragmatic aspect for the patient themselves. So I have a question for Trisha. Can you maybe explain to us some of the pros and cons, some of the benefits of getting, for an individual patient, seeking genetic testing? And what are some of the potential downsides?
Thank you. Absolutely, in terms of pros and cons of genetic testing. But just before that, the CMTRF and research is critical to improving outcomes for patients. I would absolutely want to start with that. And research progress really cannot be made without selfless participation of individuals with CMT and their family members. So absolutely start there. But in my role as a genetic counselor, talking with patients, often in terms of their immediate benefits and outcomes, research isn't at the top of the list. What might be at the top of the list is this really CMT? And can I avoid any of these medical procedures like EMGs, which I'm told really suck? And I would love this to be clinically confirmed. I want a confirmation of this diagnosis so I can move on. And you can tell me what I can and can't expect in terms of the next 4 years of my life and the next 10 years of my child's life. And darn it, is there a treatment? Is there a targeted treatment for this? Also, what I've, all these things that I've already heard you wonderful, wonderful people say today. How is this going to affect my family? Are there other people, my aunts, uncles, parents, children who might experience something similar? And of course, whether or not there could be a clinical trial that I might qualify for. In terms of, so pros, in terms of potential cons or downsides, I always ask those reasons for testing. Why are you coming in? What are your concerns? Because the possible outcomes, the testing options are going to be different based on why you're there. If a result is positive, you choose to have a genetic test and the result is positive. Positive isn't always perfect. The positive result might give you a name, which can be completely reassuring. You can be off that diagnostic odyssey. You can have more information about natural history, what to expect. So is that a benefit? Yay. Is that a drawback potentially? Because I might not be able to perfectly predict what you can expect. We've heard already from lots of wonderful people that the individuals with the exact same gene mutation or a pathogenic variant in the exact same family all have different levels of symptoms, different ages of onset, different severity of symptoms. So we always can't predict what you're going to experience. And is that potential downside to genetic testing? We don't have a crystal ball. And as we've heard, these genes don't yet have specific targeted treatments. There's a ton of work going on, and we want to get there in the next 2, 3, 5 years, but we don't have them yet. So we go back to your treatment currently is still symptomatic, even if you know the name, even if you have the gene related to this test. And then genetic testing, a lot of you, I don't know how many of you in this room have had genetic testing, but there you go.
Thank you.
Oh, yeah. And as Wolfgang said, lots of times that test is negative. That can be pretty disappointing in terms of an outcome. It can prolong that uncertainty. Oh my gosh, this diagnostic odyssey that I'm on, that can be frustrating. It can make people angry. It can cause anxiety, can be just depressing. Like, what am I supposed to do? What am I supposed to tell my family? How am I supposed to know what's next, in my life, in my child's life? What am I supposed to do to prepare? If you've had genetic testing recently, you might also have come across this result called a variant of uncertain significance, which is a possible outcome that I like to mention because sometimes it feels like a downside. But in general, I mention it because, scientists and medical researchers don't yet know every gene that's associated with CMT. A test can be negative, and you can have a variant of uncertain significance, and you can still clearly have a medical diagnosis of CMT. We all, and people have already said this, we all have variation within genes that can be benign or normal. We all have variety that can just make us amazing people, right? Those are the differences. Or like, okay, maybe might not be amazing. Cilantro tastes like soap to me and might be delicious to other people. That's, some genetic variety that I have. But there is some variation that is uncertain because we scientists, medical researchers don't know if that that gene is associated with CMT, or that change in that gene that is known to be associated with CMT can cause it. So it's like, Trisha, oh my gosh, why did I get that test? Or Trisha, I'm getting this test because of ABC. And I love to talk with people about what that can and can't tell them and why. Because each of the individual's questions and possible results have a different level of importance. And that leads individuals to different impacts related to their decision about whether or not to move forward with that test.
That's fantastic. Thank you so much for that thorough pros and cons review of genetic testing. And we'll get into the scenario in which the test comes out negative a little bit more. But of course, as you outlined, quite often the test is positive. And what I feel so excited about personally is that we are now in an era really where, at least for a couple of those genes, they are immediately actionable, right? In some sense that maybe many of the audience will know about a gene called SORD, for which this gene was only discovered, I think, 3 or 4 years ago. And we are already running a clinical trial to see whether there is a potential treatment that is efficacious. And then we heard today about DTX Pharma, about their amazing strides forward for type 1A, and so in this way, I really feel like we're moving into an area, into a time in which genetic testing is no longer what one patient described to me as meaning it means everything, but it means nothing at the same time too, because you can't do anything about it, even if the test comes back positive. We're moving into a phase in which there is something to be done about it. Okay, great. So now a question for Nicole. Let's say I, as a patient, I've been listening to this panel and I'm totally convinced I want to do genetic testing. Let's go. How do I do this? How do I go about it? And maybe one concern that I might have is, who pays for all of this?
Yeah, you got to talk about genetics, and I have to fix the whole US healthcare system. Just out of curiosity, has anybody been frustrated or had trouble ordering genetic testing? Just really, okay, yeah. Again, would love to kind of hear those talks. In the United States, medical testing is ordered by a medical professional, right? Someone with an MD license. And so to get clinical level testing, you do have to start there. I will say about 60% of my CMT testing comes from clinicians that self-define themselves as neurologists. And so that might be a good place to start with a specialist. I have 30% that refuse to self-declare. I don't know who they are. And then some pediatric specialists, some genetic specialists. I do think it's frustrating if you try to go to a doctor with something printed out from Google and say, I want this test. And I will say, hearing from the doctor's side, it's hard for them sometimes to get that piece of paper and feel a little embarrassed, and maybe they have to run out of the room and look things up. So that kind of approach sometimes will take a little bit more work. I work with a lot of genetic counselors and love them passionately. I've never seen an MD refuse to sign an order that came from a GC. So they might be some extra help for you there. But I do think that there's more accepted testing now. And the better that your phenotype or your look or your presentation fits what that doctor understands to maybe be associated with a CMT, I would say the more likely you are to have them offer or be ready to order a CMT panel. We also get a lot of CMT hits on our general neuropathy panel, or so that has a lot more genes, and that I think doctors order when they can tell there's something going on, but they're not quite sure what, and they cast a bit of a wider net. Who pays for it? I would love to get paid. And so it is something that we work on frequently also. And the answer is it's all over the place. Usually diagnostic testing, I told me I'm not allowed to say usually. It would be great if diagnostic testing was always covered. It's not, it's very insurance company dependent and the hurdles that you have to meet for your own particular company. Even if your insurance does cover it, they will pay for it. It's not unusual that it comes out of your pocket because you haven't met your deductible. And that's kind of hard for people to understand sometimes. Too, if you have a large deductible and you haven't run through it yet, even though they cover it, you can pay for it. I will say that I'm sure, I know my company, and I'm sure the majority of companies do have programs that work with patients, and I think that the GCs know about them too. And so I would encourage people, if you're feeling frustrated, if you're looking into it, call the company and ask them. We are more than happy to tell you. I'm not in sales. I'm not allowed to lead with any of this. And honestly, I try to stay out of just how much it costs. I was actually shut down from some sales visits because I offer stuff for free as a medical director, and so I'm not allowed to go anymore. But there are a lot of programs that work directly with patient pay pricing. So I would reach out, and we are devoted to getting you that information to help your clinical team make good choices. We'll really try to do that for you.
Amazing. Thank you. And forgive me for putting you on the spot with this really rather difficult question. Now, can you hear me now? I think I'm not on mute. Anybody can hear me now? Yes. Okay, there we go. So I wanted to say thank you for that question. Forgive me for putting you on the spot with this rather tricky one. Yeah, the U.S. medical healthcare system is a tough place to navigate, and this includes genetic testing, unfortunately. So you mentioned something called a panel, a genetic panel, and Trisha, in your capacity as a genetic counselor, you help patients understand what the results of a genetic test, such as a panel, really mean. Now, as I already outlined, and you mentioned it too, quite often when these panels come back, they come back negative. And then the next step is often something called whole genome or whole exome sequencing, maybe first. So these kinds of technologies patients will have heard about, but actually I'm going to see whether I can put on this one more slide to give you guys a little sense of what I mean by that type of sequencing. So a genetic panel, I hope everybody can you give me a thumbs up if you can see the slide again? Okay, perfect. So just to orient you a little bit, a genetic panel, let's see, we have here a strand of DNA and there are some genes on it, gene A and B and C, and maybe gene B is a known CMT gene. What a genetic panel will do is to sequence only a number of candidate genes or number of known CMT genes and give you back the results for those. So it's very targeted. Whereas whole exome sequencing will sequence all the genes, sort of all the genes, but most of the genes will be sequenced regardless of whether we know that they're implicated in CMT or not. And then finally, whole genome sequencing will not only sequence the genes, but also all of the other regions of the DNA. You can see here these gray bars, they align to these blue boxes. So that's sequencing the gene, whereas here on whole genome sequencing, they sequence all of this stuff. Okay, and then Nicole said we can't really, it's hard to put an exact number on what these things cost. So I just put $1 sign, $2 signs, $3 signs. That sort of captures the idea that, of course, the more you sequence, the more expensive these things are. Now, Trisha, as many patients look at this slide maybe, and hear that genetic panels might come back negative, you might ask yourself, why not do whole exome sequencing right away? Why not do whole genome sequencing right away? Let's put cost aside for now. Seems like it would be smart to just start with a more comprehensive test right away. Can you comment on that?
Well, let's do it. Okay, really. I'm going to back up a little bit because sometimes the likelihood of the result would depend on how well in each individual's symptoms match the what's known to be associated with that gene. Okay, sometimes it can be very clear. You have a family history of X and somebody in your family has already tested positive related to that gene. So I'm going to order one because it's going to be much faster and much less expensive. Or your symptoms very clearly match a gene that we already know to be associated with a gene or a gene on that CMT or hereditary neuropathy panel. But really, it is true that pretty recently some scientists and medical researchers have said whole exome sequencing might be the gold standard. They're potentially suggesting that because lots of times in conditions for which there's a lot of variability from one person to the next. We see differences in the symptoms individuals have, where multiple individuals in the same family have symptoms, but they all have slightly different features. That this whole exome sequencing might be easier to pick it up, both from the first picture you showed during your introduction, Wolfgang, and excuse me, and this one. So, hey, potentially, why not do it? The benefits being also reanalysis can sometimes happen with whole exome sequencing a couple of years later. Because new genes can be, you have a panel of those 100 genes. All right. But 2 weeks from now, medical researchers, scientists identify potentially a new gene associated with symptoms of CMT. Sometimes it doesn't get put on the panel before you have the test, but the whole exome sequencing could potentially use that data. And I mentioned reanalysis is available. And actually, sometimes I've been recently told and learned that there can be less variants of uncertain significance. So, hey, there you go. We can talk about that later. You said not to mention the cost, Wolfgang, so I won't do that. But the reasons potentially not to do it, sometimes it takes, not sometimes, it takes longer to get the answer. So if you need an answer quickly, for some immediate decision making, it also might not be the right choice for you in that moment. And it's always best if you test multiple individuals at the same time, a trio, you and your parents, because you find out if there are those de novo mutations. And if there are other people in your family who also have symptoms, you can kind of track that throughout these genes and the whole genome. So, but I do want to say no level of genetic testing is perfect. There's always another chance, even after whole exome and whole genome sequencing, that we recommend a different type of genetic test. And so it is, as everybody's already said, it's important to have a conversation with your physician and then your potentially a neurologist, a CMT specialist, a genetic counselor, to find out more and what would be right for you.
Yeah, that sounds exactly right to me. Now, from some data that I was able to capture while researching for this panel, it actually seems to me that a lot of commercial testing companies are offering and are very active in providing clinical whole exome sequencing. I'm just going to mention some statistics. GeneDx, which is one of the genetic testing companies like Invitae that provides this kind of testing, says that they have now sequenced 500,000 clinical whole exomes over their lifespan. And I think Invitae tweeted recently, or maybe you have to say X'd now, tweeted recently that you have tested over 4 million individuals, now probably not all with whole exome sequencing and probably not all with CMT, but it seems like there is a lot of testing going on. And so as I outlined in the beginning, for us to see the patterns in CMT, to find new genetic diagnoses and so on, it's important to put all that data together. So what I wanted to ask is whether you can shed some light on to what extent this kind of data, when a patient seeks genetic testing with a commercial company, is available for the biomedical research community to make progress. This is a question for Nicole, sorry.
Yeah, I'll take it. That was a lot of questions. So I will do my best. First and foremost, I do want to reiterate that I am talking about clinical testing, medical grade testing. And so in that, your privacy is of prime concern. So I don't want anybody to think that the relationship that I have with you and your doctor when you send me a sample for a medical test result is then out in the public domain. So that is something that patients can choose, and we do reach out and ask and love if people would like to make their information accessible to research. But if a patient says no, or if an ordering clinician, when they order it, says no, that is, I'm very bound by that and actually personally feel strongly about it. So I'm sorry, Wolfgang, you can't have everything. And I understand the power of it, but everyone's medical information and sample and data is their own to decide to share. So we'd love to advocate for it. It's yours. But information that I do have that is anonymized, I think can be very powerful. One of the things that we do share that you can look up yourself is a public database called ClinVar, CLINVAR, stands for clinical variant. Anytime we discover a novel variant or we find a variant that other people know about, but it's the first time we're assessing it, we do a full scientific research project on it, and then we deposit that into ClinVar. And as a company, not for CMT, but for all disorders, have had millions of deposits into that database. And I think we're actually the largest contributor currently to that. So that is available to everybody all the time. We have done 4 CMT majority panels, large panels, and I think I have over 40,000 individual CMT orders. And that is a lot of data. Our clinical hit rate, so when do I diagnose a person, is closer to 20% of the time. And I also find carriers for the autosomal recessive portions. If someone in your family is diagnosed with CMT and you know the genetic variation, family members for us, this is why they don't let me come. Family members are done at no cost, and the genetic counselors will know the other companies that do that too. So that is available. We do like to engage in research and research projects and are happy to have researchers come to us. I don't have probably a lot of the information that you think I do. I'm required to have 2 identifiers on a test requisition form. So I know people's name and I know their date of birth. And then I know their genetic test result. I will sometimes work with a physician and can go back and forth if there's a complex presentation, if they have questions, we're always happy to do that. I don't have access to your medical records, so I can't put that together. Are you okay if I speak a little to the exome question?
Sounds good to me, but maybe just a few sentences because we do want to move to the Q&A with the audience.
Oh, let's do that.
So maybe 2 sentences and then we move to Q&A.
Yeah, I was just going to say, you can think of your DNA as a set of encyclopedias, right? And so most of us are older enough to remember what a set of encyclopedias looks like. And each of your chromosomes is a volume. So when I am looking at your DNA, I'm looking for typos, right? So if I know the one gene and I know what your family typo is, that's like getting the answer key along with a test. I can really tell you quite easily, you have your familial variant or you don't. If a clinician knows that they're focused on CMT and they're ordering well-known genes, I only have to look for typos in about 60 genes on our panel. And I'm pretty good at doing that. And so what I do is I take your DNA and I use a molecular Xerox machine and I make millions of copies of just the pages that I want to look at. And that makes it really easy to find the typo. And if I gave you 100 copies of Trisha's paper here, you could find her typo really easily with one gene.
I'm sure there are no typos.
When I'm doing whole exome and whole genome, it would be like me giving you the set of encyclopedias and say, find the typo. You could probably find it. It's a lot of work and you could probably miss it pretty easily too. So I think people hearing the word whole genome, whole exome makes you believe that I know everything about it. And I don't. It's like a low pass run through and I'm looking quickly for typos. And sometimes I get a hit and it may or may not really be applicable to your disease. So it's great for research and it's great for learning things. It's not always the best if something's brand new, novel, has never been seen before for helping to guide clinical management. So again, getting the exome, getting the genome, it might be your next step, your next odyssey, and we may find something. when I get an answer back from an exome, I put it through some clinical filters and I only look at the genes that are involved in the pathways that we know CMT is involved in. And so until the field is really caught up and the research is out there, I can't even tap into anything to learn about to look in those genes. So I guess I would say that if you have your whole exome done, we can do reanalysis for a couple of years. We do it every 6 months automatically. We'll contact you if we find something. We'll tell your clinician that we found it. But I would say then in another couple of years, technology is moving so fast, somebody's going to ask you to do it again. And you'd be like, but you did my whole genome. I'm like, I did some of it. So thank you for that.
All right, so we have maybe time for 2 or so questions from the audience, if I get the okay for that. I can't see the audience, so you guys have to help me pick folks. I can't see who is raising their arm.
There's a person walking around with a microphone.
You go ahead. Thank you. Yes, so the variables that I'm curious about are the age of onset and also the difference in severity of symptoms with people who have the same categorization. And in my family, people get diagnosed in their 70s. So I'm just thinking it would be wonderful if the age of diagnosis would be postponed to about 200, and then we'd live lifelong lives without this. So I'm curious to know if the genetic testing can look at all the people who have a given genetic type and then, compare the intergene varieties with age of onset and with severity so that we can tease out how to diminish the severity of symptoms and also how to postpone the age at which they present.
We think it's a Wolfgang question. No, we're kidding. Yeah, it's a great question. So what you're talking about is sort of a spectrum, right, of things that present. And I'm going to take it in 2 pieces. One is the same variant. So same variant, same family, and you present in different ways. That's because it's not the only gene that you have, right? And so and it's very complex pathway where the problem is. And so we think if you present later or with a more mild form that you have some other variants that help to compensate or help you along. And that if you present earlier and more severely, that you maybe either don't have those compensating other factors, other proteins to help you along, or maybe you have something extra that makes it a little bit worse. So that's why we do see a spectrum, which makes it a little hard to pin down. And then in disorders like this that have a long progress, you have a lot of environmental factors, I think we would say, that could help affect.
Yeah, I think, did that answer your question? I feel like there were 2 parts.
Yes, I understand there are other genetic factors that they use.
Yep.
But can we identify them? Or can we identify environmental factors so that we can help deal with this condition before we have an absolute cure?
Wolfgang, do you want to do that from a scientific perspective? Can we identify modifiers?
Maybe I can jump in quickly. I think this question is really, really, really great. The variability that we see phenotypically for people that have the same mutation is so remarkable in CMT. One of the benefits of getting genetic testing and getting confirmed that you have this type also is that you might be able to enroll in what we call natural history studies, or studies that really track the development of the disease over time. And there, you might not only track how you're doing, but you may also start tracking, what your environment really looks like. And that sort of direction could be a path for us to identify what environmental influences have an impact on the development of CMT and its symptoms. So what I'm outlining here is sort of a scenario that really, in reality, we haven't implemented at very large scale, but it is one way in which we could try and discern what environmental factors really have an influence on CMT. And environmental factors doesn't just mean, what do you eat, how do you sleep, and so on, but it also means how do you exercise? What do your daily activities look like? And I feel like for a lot of CMT patients, including myself, the question of what should I do? What should I avoid in terms of daily activities is one that we still, as a research community, haven't properly answered. But one way to get started with this is to have your genetic diagnosis confirmed so that you can then enroll in studies in which you can try to find those environmental influences that determine the outcome, the development of the disease. So then we have, okay, I just hear that it's time to wrap up. So unfortunately, I think we ran a little out of time. Nevertheless, I hope that everybody enjoyed this panel. I certainly found it super informative. I thank Nicole and Trisha for being here. I apologize again for not being there in person. I wish you all a continued great day. Thank you to the CMTRF for bringing this community together every year again. Yeah, thanks. Bye.